So now we're going to discuss the purine metabolism clinical correlates. So we're going to be discussing the clinical applications of learning the purine metabolism pathway. So we're going to be discussing the pathologies that are associated with abnormalities in purine synthesis pathway, and we're also going to study the drugs that target the purine synthesis pathway. So we're going to start by discussing drugs that disrupt the purine synthesis. We're going to talk about ribavirin and mycophenolate, azathioprine and 6-mercaptopurine, and finally allopurinol. So the first drug that we're going to discuss is going to be ribavirin and mycophenolate. Now, these two drugs have the same mechanism of action, is that they inhibit the enzyme IMP dehydrogenase. Now, this enzyme is responsible for the conversion of inosine monophosphate into guanosine monophosphate. So the use of this drug is going to inhibit this enzyme, leading to decreased production of GMP. That's going to lead to decreased DNA and RNA synthesis. Ribavirin mainly affects viruses. That's why it's used as an antiviral drug. Mycophenolate mainly affects lymphocytes or white blood cells. That's why it's used as an immunosuppressant. Okay, so the next group of drugs that we're going to talk about is azathioprine and 6-mercaptopurine. Now, these are pretty much the same drugs. Azathioprine is actually converted to 6-mercaptopurine in the body. So 6-mercaptopurine is a purine analog, so it has a very similar structure to hypoxanthine and guanine. And that gives it the ability to interact with PRPP by the enzyme AGPRT, and it forms a substance called thioionosine monophosphate. Now, this is a toxic substance. So what it does is that it inhibits multiple enzymes in the de novo purine synthesis pathway, and it's going to prevent the formation of IMP, GMP, and AMP. It's going to prevent DNA and RNA synthesis inside the cell, and it's going to lead to cell death. So these drugs can be used as cancer medications and also as immunosuppressants. One of the pathologies that could arise from abnormalities in purine metabolism is gout. Now, this is excess uric acid in the plasma. So the excess uric acid deposits in the crystal form in joints, and that can lead to pain, swelling, and also redness. It could be caused by overproduction of uric acid or decreased excretion of uric acid. And in these cases, basically the treatment of gout is going to be to give a drug called allopurinol. Now, this is a drug that inhibits the enzyme xanthine oxidase, and when you inhibit this enzyme, you're going to decrease the production of uric acid. An important drug interaction that you need to know is between allopurinol and azathioprine or 6-mercaptopurine. Now, azathioprine, when ingested, is going to be converted to 6-mercaptopurine. 6-mercaptopurine is going to be converted into its inactive metabolites by the enzyme xanthine oxidase. Now, remember that allopurinol inhibits xanthine oxidase. So if a patient is taking both of these drugs together, this can increase the risk of 6-mercaptopurine toxicity. So be cautious when giving these two drugs together. Okay, so the next pathology that could be seen with abnormalities in the purine metabolism is something called Lesch-Nyhan syndrome. Now, this is an autosomal recessive disease in which there is absence of the enzyme AGPRT. Now, if you recall, this enzyme is responsible for the purine salvage pathway. So it's responsible for taking guanine and hypoxanthine and converting them back into their respective nucleotides. So if you have a deficiency or absence of this enzyme, that means that this patient is going to have excess uric acid production. This is why these patients usually present with juvenile gout. They could also present with neurological impairment. The mechanism here is not very clear, but they can present with hypotonia, chorea, and something very characteristic of Lesch-Nyhan syndrome is self-mutilating behavior, like biting and scratching themselves, as you can see in this picture. The last pathology that we're going to talk about that is associated with the purine synthesis pathway is called adenosine deaminase deficiency. Now, this is an autosomal recessive disorder in which there is a deficiency or absence of the enzyme adenosine deaminase. Now, this enzyme is very important in the degradation of AMP. So it's responsible for converting adenosine into inosine, and then inosine will be eventually converted into uric acid for excretion. So patients who have an absence of this enzyme are going to have accumulation of adenosine in their cells. Now, the most susceptible cells to accumulation of AMP is going to be the lymphocytes. So adenosine deaminase deficiency is actually one of the causes of something called SCID, or severe combined immunodeficiency, because basically it's going to lead to dysfunctional lymphocytes.